a professor, an academician, or somebody in a similar profession? Are you looking for a research journal to get your research paper published which can benefit the nation? If so, then your search has ended, because I am here to tell you about TEJASVI, which is a research journal registered by RNI for registrar of Newspaper of India, and ISSN, which is International Standardized Serial Number. To get your research paper published with TEJASVI, which is a registered magazine with Government of India, you can log on to our website www.tejasviaspitra.com, or you can also send us an email at editor.tejasviaspitra@gmail.com. You can also get your article published with TEJASVI web portal, which you can find on our website www.tejasviaspitra.com. Am I audible now? I think
Yes,
my
ma'am.
mic went mute. All right, so I was just telling, again, I was doing a little housekeeping here. Mr. Srivastava covered a lot of things, but I would like to request a lot of to all of you, please keep your mics mute when you are not speaking, especially when there is a speaker who is speaking. Please turn off your video camera if you have to leave the frame for any reason, be it eating something, drinking something, or you have to attend something else. Do not leave the meeting. I see a lot of people are leaving the meeting and then joining back again. Just turn off your video and put your mic on mute. At the end of each speaker's presentation, you will be given the time to ask them questions. So you can unmute yourself and then you can ask your questions then at that time. And this whole webinar is being telecast on the DN News channel live, which is on YouTube, and they are our official media partners. Time to call our chief guest for today, Dr. D.K. Satsangi, who is the head department of cardiothoracic and vascular sciences in All India Institute of Medical Sciences, India. He's here with us to enlighten us on various issues of research methodology. I would request Ms. S. Padmavati to please introduce Dr. D.K. Satsangi.
Yes, ma'am. Yes, ma'am. Am I audible, ma'am?
Yes, you're audible. You can please go ahead and introduce Dr. Satsangi.
Yes, ma'am. Good afternoon to all. A hearty welcome to the International Webinar 2020 on Research Methodology, Trends, Quality, Ethics, Value, Maharaja College, Vizianagaram. I am S. Padmavati, lecturer in physics, Maharaja College, Vizianagaram. I am fortunate to introduce an eminent person, Professor Dr. Deepak Kumar Satsangi. Medicine bridges the gap between science and society. Indeed, the application of scientific knowledge to human health is a crucial aspect of clinical practice. Doctors are the one important agent through which the scientific understanding is expressed. Keeping in view the importance of a doctor to the society, our organizing committee of the current webinar has chosen a best person of the field, Dr. Deepak Kumar Satsangi.
Thank you, Ms. S. Padmavati. Dr. Satsangi, can you hear us? Yes, sir. It's perfect now. And you can begin with your presentation, sir.
Okay. Now, at the outset, let me thank Devendra Srivastava for inviting me at such an important webinar program and on research methodology. Now, what I personally feel that he is doing a very good job by organizing such useful webinar programs. And earlier he invited me to participate in a program on the COVID-19, that is the coronavirus, which was very useful and very pertinent to our present situation. This is another very important subject of research methodology. And I, being a medical person, I will only touch the medical part of the research, that is research methodology in the medical research. And as you know, I will just go on my slide presentation so we can move together in an audio-visual manner. You are not seeing my presentation?
No, sir, we are still unable to see you. Yes, we can hear
Again,
you.
good afternoon, everybody. Now, without wasting much time, we should go for the topic of discussion today, and that is research methodology in medical research. Now, what is research? Research is a logical and systematic search for new and useful information on a particular topic. Research is a planned activity leading to generation of informations that will help in answering our specific questions. Now, what are the types of research? Usually we talk of two types of research. The conventional research includes descriptive studies and analytical studies. The unconventional research that includes, which is gaining more and more importance nowadays, includes operational research, evaluation of health systems, economic studies, which are the cost benefit, cost effectiveness, etc., qualitative research, and the research synthesis, that is reviews and the meta-analysis. Meta-analysis is when you analyze the data from the multiple studies. Now, what are the objectives of research? These are very basic things we have to discuss. The prime objective of research are to discover new facts, to verify and test important facts. Number 3, to analyze an event or process or phenomena to identify the cause and effect relationship. Number 4, to develop new scientific tools, concepts, and theories to solve and understand scientific and non-scientific problems. Number 5, to find solutions to scientific, non-scientific, and social problems. Number 6, to overcome or solve the problems occurring in our everyday life. So these are the objectives of the research. And what is the basis of the research methodology in medical research? Let's see that every patient in medicine is different in the way the disease manifests and also in the response to treatment. An effective treatment for 90% of the population may not work for the other 10%. Thus, medicine is said to be inherently experimental. Even the most widely accepted treatment need to be monitored and evaluated to determine whether they are effective for specific patients or for patients in general. This is one of the functions of medical research. Now let's see what is the basis of the research methodology in medical research, and that is even the best proven prophylactic, diagnostic, and therapeutic methods must continuously be challenged through research for their efficacy, accessibility, and quality. Number 2 is another function is the development of the new treatments, especially new investigational drugs, medical devices, and surgical techniques. In other words, the purpose of medical research involving human subject is to improve prophylactic, diagnostic, and therapeutic procedures and understanding of the etiology and pathogenesis of the disease. So we see that these are the ongoing process. Now, what are the steps in medical research? This is very important. Number 1, to formulate a research. Number 2, to identify the problem, what problem we are going to study. Number 3, formulating a research question. Number 4, refining the research question, that is review of the literature. Number 5, formulate hypotheses and research objectives. Number 6, decide the study population and setting. Number 7, decide on the study design and methodology. Number 8, writing the protocol. Number 9, collecting the data. Number 10, analyze the data and apply statistical significance. Number 11, write the report. See, I will again repeat because this is very important, and these are the points we are going to elaborate in this presentation. Number 1, to formulate a research. Number 2, to identify the problem. Number 3, formulating a research question. Number 4, refining the research question, literature review. Number 5, formulate hypothesis and research objectives. Number 6, decide the study population and setting. Number 7, decide on the study design and methodology. Number 8, writing the protocol. Number 9, collecting the data. Number 10, analyze the data and apply statistical significance. And number 11, write the report. Now, identify the problem. Now, number 1, most important is interest and expertise. The topic should be interesting to the investigator, to the funding agency, and the medical community. Number 2, it should be relevant and applicable. So, research should be new information to the scientific society, or expected result is likely to alter clinical decisions in future. Then feasibility, it should be feasible in terms of time, manpower, and money. These are very important to decide before we embark on the research. Now, number 2 was formulating a research question. Research question is a formal statement of the goal of a study. Foremost among these is whether the question is interesting. Then it is important that the investigator is genuinely curious about the question being investigated, so that he or she can remain motivated till the successful completion of the study. Curiosity is also an asset in terms of stimulating questions for the future study. So we see that curiosity is the key factor in the entire research. Now again, formulating a research question. Next is feasibility. It should be feasible. Number 2, the third consideration is the novelty factor, or the potential of the study to contribute something new to the knowledge base. This is very important. Related to novelty is the relevance of the research question, and it should add to existing knowledge, guide future studies, or have implications for education, clinical practice, or healthcare. So it should be very useful research, and when we complete it, it should be useful to the society, to the medicine, and to the population at large. Now again, formulating the research question. Finally, the idea must be ethical. It is very important. The studies that invade people's privacy or create possible physical or psychological risk are ethically unacceptable. So as you see here, a good research question is, it should be non-biased, it should be answerable, it should be relevant, it should be intriguing, it should be pertinent, and it should be focused. So these points you must satisfy before we evaluate our research question. So a good research question could thus be described by the acronym. This is the FINER. So F stands for feasible. The question should be feasible. It should be interesting to the investigator. It should be novel. It should be ethical, and it should be relevant. Now, a useful format to use in the development of a specific research question is again by the acronym PICO, where P stands for the population of interest, I stands for the intervention or the treatment being studied, C stands for the comparison with other group, and O is the outcome of interest. Often timing is added to the PIC, indicating the time frame in which the study will be completed. It is very important. Other, if it is not completed in time frame, the research will be obsolete or your funds may get exhausted. In patients with pneumonia, whether treatment with X, that is I, intervention, compared to Y, that is comparison, reduces the number of days of the hospital stay. So this is the PICO system. Again, anatomy of a good research question is, PICO is a focused clinical question that consists of 3 or 4 of these concepts. Writing a PICO question facilitates the search process and the identification of best evidence. Now, for example, PICO example 1, as we say, with a comparison C, this is a comparative study. For example, in a 30-year-old male patient diagnosed with depression, that is P, and is the St. John's wort, that is intervention I, is as effective as SSRI, that is the comparison C, for reducing depressive symptoms. So that is the outcome, and that is the example of PICO. Now, another PICO example 2 is without a comparison, where C is not there. In adults with back pain, does yoga therapy reduces pain? That's the outcome. So here is no comparison. This is known as one of the deductive studies. Now, third point was refining the research question. That is, review of the literature is very important. And review of the literature concerned to our research project is, once the problem or question is specified, the next step is to collect as much related information as possible. Literature review will help to determine to what extent the issue or research question has been previously researched, to identify the past relevant studies as well as the methods used, to refine the research question, and also to put the project and methodology into a relevant concept. So it is very important. We should know our background, we should know what have already been searched, and we should know that how this information is going to help us in doing our research. Now, number 4 was to formulate hypothesis and research objectives. The research hypothesis is developed from the research questions. For example, in the research study, comparing treatment X versus treatment Y in patients with pneumonia, the experimental group would be treatment X and the control or conventional group will be the treatment Y. So we have the 2 groups and we have to compare the efficacy of our intervention or treatment. Now, formulate hypothesis and research further, we see the investigative team would first state a research hypothesis. This could be expressed as a single outcome. For example, treatment X leads to the improved functional outcome. So that is what we see here. These are the various types of the hypothesis, which may be deductive, which may be inductive, which may be simple, which may be complex. The deductive may be further statistical or may be alternative research. Number 5 was decide the study population and setting. What is the material method? On which kind of patients we are going to do a study? And the definition of the subject of a study and the target population should be clearly split out. Whether the inclusion and exclusion criteria, what type of population is included, what type of population is excluded from the scope of research. The sample size is very important, as we see later on. The smaller the sample, the more will be the uncertainty. If we are going to do research on a very small number of patients, there will be uncertain results. So sample size should be chosen in such a way that the finding in the study accurately reflects what is going on in the population. So this is very important. Then decide the study population and setting. A well-designed study, poorly analyzed, can be rescued by reanalysis. But a poorly designed study is beyond the redemption of even sophisticated statistics. So it's very clear that study the population setting is very important for any research. Now, the study population again, to get valid and reliable answer to the questions, appropriate research design and method is the prerequisite. Study design is the framework in which investigation is planned and carried out. Selection of design is necessarily based on type of research question. So again, we repeat, to get valid and reliable answer to the questions, appropriate research design and method is a prerequisite. Study design is the framework in which investigation is planned and carried out. Selection of design is necessarily based on type of research questions. Now, what are the research design? This was the 6th point. The observational studies in which subjects are observed include the case study or a case series. This may be a case and control study. This may be a cross-sectional studies, or this may be the cohort or the longitudinal studies. Now let us take them. And number 6 is the research designs. 6th point is research designs, whether experimental studies in which the effect of an intervention is observed in controlled trials or diagnostic tests. Now research study designs. Now we see, as we have already learned, that the number 1 is the case reports, a single case, or a case series, a series of cases, or analysis of secular trends, or the case control studies, as we see the case study of case along with control, or cohort studies, where we just find that a group of patients is specific in which we have to conduct the studies, or randomized trial, clinical trial. This is also very important. If we cannot study the whole population, we randomize, pick up the samples from different places, and then we study. Now, among the case report, number 1 are simply reports of events observed in single patient, useful for raising hypotheses about drug effects, leads to the drug test with more vigorous study design, very rare to use to make a statement of causation. Number 4 exception to this is when the outcome is very rare and so characteristic that one knows that it is due to the exposure. And number 6 is accepted when challenge situation is very fatal. So this is about the case report. If you are studying a single case, we should keep these points in mind. If you have a case series or a group of patients, collection of patients, all of whom have a single exposure, whose clinical outcomes are then evaluated and described. Alternatively, case series can be collection of patients with a single outcome, looking at their antecedent exposure, useful for quantifying the incidence of an adverse reaction or whether occurs in larger population, and just provides clinical description of a disease or of patients who receive an exposure. So if we do the case series or a group of patients who have received exposure, this is known as the study of case series. Then analysis of secular trends, this is called as ecological studies, examines the trends in an exposure that is presumed cause and trends in a disease that is a presumed effect, and test whether the trends coincide. Vital statistic and record linkage are often used in these studies, useful for rapidly providing evidence for or against a hypothesis, unable to control confounding variables. For example, lung cancer might be the cause because of cigarette smoking, but chance of occupational hazards can still not be ruled out. So this is the secular trend, These are certain problems, but it will be helpful in deciding whether a particular study is going to rapidly provide the evidence for which we are searching in our research. Then is the control case and control studies, as you say, 2 groups. One group, which is the clinical cases, other is which is controlled, not exposed. So compare cases with the disease to controls without the disease, looking for differences in exposures. Multiple possible causes of a single disease can be studied. Helps in studying relatively rare disease, requires a smaller sample size. Informations are generally contained retrospectively from the medical records by interviews or the questionnaires. Limitations are validity of retrospective information, and selection of control is challenging task. The inappropriate control selection can lead to incorrect conclusions. So one has to be very, very cautious while collecting the control group and also the case groups. Now, cohort studies, as we know, certain definite group identify subsets of a defined population and followed them over time, looking for differences in their outcome. Used in compare exposed patient to unexposed patient, can also be used to compare one exposure to another, or when multiple outcomes from single exposure is to be studied, either done prospectively or retrospectively more reliable causal association, but requires large sample size even for an uncommon outcome and can require prolonged time period to study delayed outcomes. So cohort studies, they are going to give us the best results, but there are certain difficulties and one has to study a large number of population over a large period of time. So this is showing the differences between cohort study and case control study. As we can very see, the case control study disease, number one, the case where it is present, and number two is the control where it is absent. Well, cohort studies has a factor which are the presently exposed and unexposed group of the patient. Now, randomized clinical trial is very important and very useful. An experimental study, the investigator controls the therapy that is to be received by each participant. Major strength is the randomization. Problems might include the ethical issues and are expensive. They are not of big importance after marketing. Once it is marketed, the randomization is not of big importance. Meta-analysis studies, as we know, the meta-analysis is the analyzing the results and the studies of the different multiple studies and then coming to a conclusion. Meta-analysis has been defined as the statistical analysis of a collection of analytical results for the purpose of integrating the findings. The use of meta-analysis identifies sources of variation among study findings to provide an overall measures of effect as a summary of those findings. Meta-analysis is most often used to assess the clinical effectiveness of healthcare interventions. It does this by combining data from the two or more randomized controlled trial. So we have to see whether the results obtained by our research are duplicated or they are supported by the results of the other studies from good center. But when we are choosing the other studies, we should be careful. So meta-analysis of trials provides a precise estimate of treatment effect, giving due weight to the size of the different studies included. Important studies chosen for inclusion, the meta-analysis must be sufficiently similar in a number of characteristics in order to accurately combine their results. It's very important that those studies have also been done under the similar condition and on similar population with similar methodology. Now, where does meta-analysis fit in the research process? This is a very pertinent question. The graphical elements of the meta-analysis, such as the forest plot, for example, provide a mechanism for presenting the data clearly and for capturing the attention of the reviewers. Some funding agencies now require a meta-analysis of existing research as part of the grants. So application to fund new research. If you are looking for a funding agency, most of the funding agency wants that same research should be done at multiple centers under similar circumstances. So their results may be compared and a conclusion may be drawn. Now, evidence-based medicine, nowadays we hear a lot about the evidence-based medicine. So EBM is an approach to medical practice that uses the results of patients' care research and other available objective evidence as a component of clinical decision making. So this is very important. If we are doing some treatment, we want for the evidence. What are the experimental evidence, whether this intervention or this treatment is really helping the patient. So if we compare this, now this is based on the Oxford Center of the Evidence-Based Medicine. There are the level of evidences. As we see here, the level 1 is the case study, which is restoring the expert opinions, and level 4 is the case series. So case study, case series, level 3 is case controlled studies, and number 4 is the number 2 is the cohort studies, and then level 1 is a systemic review randomized controlled trial. So this is how it has been put in the brief. And then need for evidence-based medicine is very important. The daunting number of diseases, availability of broad number of therapeutic options, to keep ourselves updated in the field of expertise, information sources, to remain competent throughout the career. So this is very important, evidence medicine, because particularly in India, everybody will come with his treatment and there are hundreds of opinions. Somebody suggests some treatment, somebody suggests some treatment. There's a homeopathic treatment, there's Ayurvedic treatment, there are herbal treatment. And then we should ask, what is the evidence that this treatment is definitely helping the patient? Now, if we have the evidence medicine, we will save many patients from taking the wrong treatment or losing the valuable time. Now, writing the protocol was the 7th point, most important. All the effort put into the preceding steps culminate into the draft of the research protocol that incorporates all the information regarding the research in a concise manner. The protocol should contain background information on the study, objectives, it should include ethical aspects, it should have study design, study procedures, method of assessment, statistics and evaluation, administrative issues and references. So while writing a protocol, we should be very careful about these points, particularly the ethical aspects. Further, in writing the protocol, once the protocol is ready, approval from the ethics committee should be obtained before the start of the study. Along with the protocol, the informed consent form and other documents required should also be submitted to ethics committee for approval. Now, number 8 was the collection of data. This is very important. Once the protocol is finalized, data should be collected. The data forms should be legibly filled and they should be fully completed. Ethical issues must be taken care of from the beginning to the end of the study. In drug trials, care must be taken to document the details of adverse events. Any proper documentation throughout the study is important to ensure credibility of data. So this is the collecting the data, studying the data, then documentation of the data, reporting the adverse reactions during the drug trial, ethical issues. These are very important for the data collection. Now, analyze the data and apply statistical significance. Data should be scrutinized for internal consistency and the external validity. Data should be analyzed using the already decided data, the management plan. Now comes the important, write the report. Once we have collected data, we have analyzed data, and we have to write the report. The report should be sufficiently detailed that can remove any doubt a reader might have about any aspect of the results. It should be properly worded, should be adequately illustrated by charts or diagrams or tables which enhance the clarity. All the limitations need to be described openly. Now, most important of all, as we said, the ethical consideration in medical research. This is the most important and most priority conditions. So definition of scientific misconduct. Many people are very, they want to have an instant glory, instant success, a self-glorification. They will compromise with their studies and come out with something and advocate something which may be very harmful and fatal for the society. So most of this misconduct are scientific misconduct in fabrication, the falsification, or plagiarism in proposing, performing, or reviewing research, or in reporting research results. As you know, plagiarism is taking somebody else's idea, somebody else's work, somebody else's data, and then fabrication to fabricate in order to, particularly some companies, there are some company-supported researchers. They want the doctors or the researcher to fabricate the data in favor of their products. And with that, they make a lot of money. But who gets harmed is the poor patient and society at large. Now, there are some importance of basic ethics result. Dishonest, fraudulent, or unethical researchers can circumvent the scientific methods. The notable examples are the Nazi Germany experimentation, charges brought against 23 German physicians in the Nuremberg, and what they studied the effect of freezing on the patients. Many patients were freed to almost 0 degrees centigrade and they died. The malaria experiments, without treating the malaria, they let the patients suffer and they studied them. High altitude experiments led to the death of many of the Jews, the patients. So this led to the development of Nuremberg Code, which has very infamous code. Then we have the Tuskegee syphilis study, and this was a study conducted on the AA males in American country,
and decision was made to do long-term prospective study and follow long-term effects of the syphilis until death. And therefore, though the treatment for syphilis was available, the penicillin had been discovered, but these patients were denied the treatment. They were allowed to suffer with the disease till their death so that the scientists can study the long-term effects of the syphilis. And this is again unethical practice in research. So regulation of research and protection of the research participants, proponents of situational ethics argue that no general rules can be applied to all situations. Each action is unique. Belmont report serves as a fundamental document for the federal regulations for protection of human subjects. There are three principles. Number one, there should be respect for persons. Number two, the person should be benefited by the research. And number three, the justice should not be compromised. So CRUD requires that protocols involving human subjects be reviewed by an IRB. RB, that is the research board. Now, what is this research boards? Before that, we see that it is very complementary and supplementary and important to have an informed consent. This is inherent to this principle are four elements. Number one, subjects are made fully aware of the nature and purpose of the research and project. Number two, consent is voluntarily given. And number three, the person involved has the legal capacity to give consent. Number four, the responsibility for obtaining consent rests with the researcher. Sometimes because of the Hawthorne effect, it may be necessary to use some deception in telling subjects about the study that we can discuss later on. Now, the privacy and confidentiality in research is very important. Privacy refers to capacity of individual to control when, what conditions others have access to their behaviors, beliefs, and values. Confidentiality refers to linking information to a person's identity. Can you think of examples where confidentiality would be important? Informed consent should indicate how researchers will protect confidentially of the participants. So now again on privacy and confidentiality, some procedure that can ensure confidentiality to that person who is being researched is obtaining anonymous information, number one. Number two is all information is anonymous. We do not know the patient. Number two, code data so that identify info is eliminated. Number three, substitute other names. For example, the other name may be substituted for his name. And then do not release or report individual data. Limit access that could reveal individual identity. Report data only in group form. Use computerized method for encrypting data. So there are the scientific dishonesty is again an important factor. There may be 6 areas for the scientific dishonesty. Plagiarism, as we said, the using the ideas, writings, and drawings of the others as our own. Fabrication and falsification, making of for the altering data. Non-publication of data, also called the cooking data, the data which we are cooking. And then faulty data gathering procedures. These are all. Then we have got 2 more, that is poor data storage and retention and misleading authorship. Who should be an author of this? Sometimes they try to include so many names who has not really worked for this project. And for example, technicians do not necessarily become the joint author. Authorship should involve only those who contribute directly and discuss authorship before the project. Now, constitution of the board, the research board, this consists at least 7 members who collectively have the qualification and experience to review and evaluate the science, medical aspects, and ethics of the proposed trial. Number 1 has to be chairperson who is appointed, who is from outside the institution. Number 2 is 1-2 basic medical scientist, that is clinical pharmacologist. Number 3, 1-2 clinician from the same institution. Number 4, 1 legal expert, 1 social scientist, 1 philosopher or ethicist, 1 layperson from community, and number 8 is member secretary, which is also appointed. So as we see the quorum of the research board for reviewing and making decisions on each protocol The quorum of IRB should be at least 5 members with the following representation: basic medical scientists, clinicians, legal experts, social scientists, and layperson from the community. Now, in any case, the IRB must include at least 1 member whose primary area of interest or specialization is non-scientific, at least 1 member who is independent of the institution, trial site. Besides, there should be appropriate gender representation on the IRB. If required, subject experts may be invited to offer their view. Further, based on the requirement of research area, for example, AIDS or genetic disorders, etc., specific patients may group also may be included. So we can conclude that research is a scientific method used to collect and analyze information to increase our understanding or solve issues on a particular field. So research is a scientific method used to collect and analyze information to increase our understanding or solve issues on particular field. The research topic should be feasible, interesting, novel, ethical, and relevant. The ethical consideration should be taken care of conducting the research. So this is very important. Number 1, that research should be useful and scientific. Number 2, that it should be feasible, interesting, novel, ethical, and relevant. Number 3, the ethical consideration should be taken care of conducting the research. Number 4, the research results should not be based both the negative and positive results. So these are the conclusion, and so I will again say that it's very important, very critical issue to carry out the research, because one should not so let me see if I can switch on the video.
Thank you so much, sir. It was an honor for us, and it was a very, very knowledgeable session. And now I would like to ask all the participants if they have any questions for Dr. D.K. Satsangi, they can ask it.
Yeah, I can't see on video trying to do it. But anyway, the questions may be asked, and I'll be happy to answer without video.
Sir, very good afternoon, sir.
Yes.
We feel it's a very nice talk to hear. Sir, I have a small doubt.
Yeah, please go ahead.
Actually, I am a nuclear physicist.
Yes. You carry on, please.
Am I audible?
Stop in between. I can hear you.
Sir, actually, I am a nuclear physicist. I want to preserve some medical samples like blood and urine and sputum samples for kidney patients. So due to COVID-19 pandemic, I am unable to prevent in the medical colleges. So is there any suggestion from your side to prevent those samples for 1 week or 10 days in my home?
Can you see me on video now?
Yes, sir, we can see you on video. Yes, sir, we are able to see you now. You can respond to the question, sir.
Yeah, you can see
Yeah, you can see me and you can hear me also.
Yes, sir, we can hear you also.
Yes,
Hello,
now
can
you speak.
Can you see me on video?
Yes, we can see you. You speak out, please.
No, I could not get the question.
The question is that Mr. Suryanarayan is asking you, he is
Yes.
unable to preserve the samples of blood, urine and sputum during the COVID-19 period. He is unable to take this to the laboratory, so he wants to know, is there any suggestion from your side, any method that he can adopt to preserve these samples in the house for at least a week? Can you answer his question?
Yeah, sure. Why not? This is a common problem. The transportation of sample, should be immediately done. And there are the containers which have the eyes. So sample is collected and within 24 hours it should reach to the center of studies. Sometimes it is not possible, then it has to be, collected and preserved in a refrigeration at a temperature of 4 degrees or so. And like that, one can keep them for 24 to 48 hours. But if it has to be further kept, then they have to be put in a freezer, But it should not be delayed more than a week or so. Then definitely, if you delay and keep the samples for longer time with you, your results will be affected, and the false negativity, results will be more. So it is advisable, one should be careful that they are in a position to transport the samples immediately, and only then they should take samples. But if it is not possible, then they should preserve, in a cold 4 degrees centigrade temperature, keep it for some time, and then transport whenever it is possible for them. Does it satisfy to Mr.
Suryanarayan? Mr. Suryanarayan, was your question answered, or do you want to ask any something else?
Okay, I think Mr. Suryanarayan is satisfied with the answer.
Sir,
is there
actually,
anybody else who wants to ask anything?
Yes,
Dr.
sir.
Satsangi can ask, please.
Sir,
Can we
actually,
expect questions from Dr.
if you
Satsangi, because
preserve for 1 week
we have been
or
able
more
to
than 1
catch
week?
hold of him and bring him here
Yes, sir. Yes, sir.
with great difficulty. So make
Hello?
use of his experience and knowledge.
Yes, sir. Yes, sir.
He is head of the department in All India Institute of Medical Sciences, which is one of
Yes,
the
sir.
world's topmost medical institutes. If you want to ask him anything regarding academic area also, he will be able to answer. Nobody?
Sir, respected doctor, I have a doubt, sir. I am Dr. D.B.
Yes, sir.
Murthy. Just I have a doubt.
Yes, Dr. Radhakrishnan Dwarka.
Actually, I am PhD. I am not a professional doctor.
Yeah.
So regarding
Yes,
methodology, research methodology, in what way the research in medical field is different from research in basic sciences?
You see, the research in the medical science is in a lot of ways different from the research in other fields. The number one most important, that the end result of a failure of treatment in medicine is often death. So one have to be very careful for research, and they have to be very serious and particular about doing research in medical field. And they should know if the research is wrong. It will take not only one life of 1 or 2 people, but most of the people at large. Number 2, it has also been seen some people have not studied a particular medicine, a particular operation for a sufficient long period. They are in a hurry to introduce into the clinical practice in order to get applause or in order to get glorification. But after long term, when this has already been practiced over a long period, it was found that it was doing actually more harm to the people, patients and population at large, than doing it any good. And after that, these treatment or these drugs, they have been banned because they found that it was not the true treatment for the patient. So this is also very important. Number 3, as said, that there is a lot of uncertainty in the medicine. For example, as I mentioned, that one treatment or drug may be helpful to the patient, but at the same time for the other patient, it may be very deleterious or fatal. Therefore, nobody can say, for example, an injection of penicillin can cure a disease in a patient, but in another patient it may produce severe anaphylactic reactions and we may lose the patient. Therefore, medicine is very, I must say that it's a very imperfect science and it's still in infancy. and most of the researchers may not come out with a perfect solution for a disease. And because of these difficulties, because when you are doing research on the other branches, then you think it is not going to affect somebody's life. Do you want me to stop, Devinder? Okay. Hello. Yes.
Yes, sir. So, Dr. Radhakrishna, was your question answered?
Respected doctor, I have another doubt. Respected doctor, I have another doubt regarding the COVID. So in the social media, news is prevailing that already some medical laboratories, they found medicine for COVID.
Yes.
How far it is true?
Hello, I could not get your question. Devendra, would you translate? Devendra, would you translate the question of Dr. Radhakrishnan?
Yes, sir. Dr. Radhakrishna, can you please ask your question again? Even I was not able to understand you.
Hello, Devendra?
Respected doctor, in social media, there are so many things prevailing that some private laboratories or some medical laboratories found vaccine for COVID. They are announcing in coming 2 months we are going to introduce that medicine in the market. How far it is true?
Well, now government has recognized some laboratories and have given them a responsibility to do the COVID test, and those are doing tests up to the satisfaction of government. But certain cases have come, certain laboratories were giving false positive and false negative results, and some doctors have been arrested, some laboratories have been also closed, their license have been cancelled. So this sort of human practice is going on, which is to be condemned and which is very deplorable. I agree with you. But then human factor is there, and we have to fight against that.
Thank you, doctor. Thank you.
Thank you very much. Again, once again, I take opportunity to thank you. And I know that you are an excellent journalist, and you have all the qualities of being the very, very normal and #1 journalist in the country. You have the right ideas, you have the right qualities, you have right capabilities, and I wish you all the best to take all these relevant and problems which are important for our country, and wish you all the success. Thank you very much.
Thank you very much, Dr. Satsangi. I am really grateful that you have spared such precious moments for us, and you have enlightened us with so many great things that we really did not know. Dr. DBRK Murthy, I would like to tell you one thing, that please never in your lifetime believe in social media. These people give all fake news. They will all misguide you. That is why Government of India has come up with some new law that the social media has to be properly controlled. Social media is flashing such news that people are enticed, and they are giving so lucrative things. You will be enticed and you will fall in love with the social media, but it is not true. There are very few number of people who will give you correct picture, so don't believe in that. And yes, of course, Dr. Satsangi, whatever he has told is the right thing. And I would like to tell you one thing about Dr. Satsangi, that in his 40 years career, he has had performed about 25,000 open heart surgery with a mortality rate of 0.1%, which is very, very difficult. And he is at this age also, he feels young and he wants to do something in the cardiac field. So whatever problems you are having relating cardiology, cardiac problem, you can contact us on our email ID. You send your messages. We can forward the same to Dr. Satsangi, and he can give you suggestions, and whatever way he can help you, he will help. Thank you very much, Dr. Satsangi. for being with us. Anybody else wants to ask anything? I would like to request anybody, if you want to ask anything, you can ask.